Good evening, ladies and gentlemen. Welcome to the Novartis Lecture here on the campus in Basel. It's actually very exciting to see again a full house, almost full house. People are still coming in. It was exactly 7 years ago, December 2012, when we had the first of these lectures here in Basel. December 2012 was the month where the ancient Mayan calendar said that the world will end. We are still here, and what is even better, we have the same speaker here than we had 7 years ago. So also Professor Botond Roska is still alive. Forgive me. When we introduced Professor Roska in 2012, we said, and I quote here, that he's confident that he's on the right track towards at least partially overcoming blindness, and that he is working on a medical revolution. I think now, today, it's fair to say that this revolution has advanced. 2 years ago, Professor Roska, together with his colleague, and I would say friend, Professor Hendrik Scholl, created the Institute of Molecular and Clinical Ophthalmology here in Basel, the IOB. And they're really working very hard to make that vision for the blind, which they're going to talk about tonight, a reality. It's a huge proposition, and it's exciting to have these two gentlemen here tonight to talk about it. Novartis, just as a side note, is a co-founding member of the Institute. We are very proud that we could do that. And our chairman, Dr. Jörg Reinhardt, in a minute will share with you why we think this is an important step in science and probably also in the well-being of mankind. I'll use now big words, but it's really, really exciting. Before we come to that, quickly a few words to the two speakers. And forgive me if I have to read a little bit, but it's really, they have such a long list of things that they have achieved already. So Professor Roska is a professor at the University of Basel, both in medicine and in science. He received his MD degree at the Semmelweis Medical School in Budapest, and he completed his PhD studies in neurobiology at the University of California in Berkeley. In 2005, Professor Roska actually came here to Basel, joined the Friedrich Miesser Institute, and in 2016, the Friedrich Miesser Institute. He received a number of awards. I won't go into details here, and he authored more than 80 articles in peer-reviewed journals. His partner in crime or in research, and hopefully successful research, Professor Hendrik Scholl, is professor and chairman of the Department of Ophthalmology at the University of Basel, and he is a adjunct professor of ophthalmology at the Wilmer Eye Institute at the Johns Hopkins University in Baltimore. He graduated at the medical faculty in Tübingen and also holds a Master of Arts from the Faculty of Philosophy of the same university. Very broad. He worked in Bonn for a long time in various positions, and in 2010 was appointed professor of ophthalmology there. In 2011, he got the chair in Baltimore, and in 2016, and that's where I made the mistake before, apologies, he joined the University of Basel. Also, Professor Scholl authored many articles, 150 to be precise, in peer-reviewed journals, and also he received more prestigious awards as well. So I think you would agree that this is a couple that can really bring the research on blindness, on vision, to the next level. So I will pause here and give the floor to Jörg Reinhardt to introduce. Thank you very much.
So, guten Abend, meine Damen und Herren, liebe Kollegen. Good evening, everybody. It's also for me a real pleasure to welcome you tonight to this presentation by Botond Roska and Hendrik Scholl. It's a special pleasure because, as Mr. Yagi just pointed out, it is now 2 years ago that Novartis, together with the University of Basel and the University Spital of Basel, and with some financial support by the city, by the canton, did found this institution. I think when you look back over 2 years, a lot has happened since then. Much more is going to come. But I think we can say that the institution so far has been very successful. And I think it probably is making sense to point out in a few sentences in 2 minutes or so what the reasons were why we decided at that time to invest in an institution like this together with the university and, as I said, also the city. Now, it is an example for a public-private partnership that so far works well. That is not that often, well, that public-private partnerships are actually being created in Switzerland or in Basel. And I think all the founders are very proud of the progress that the institution has made so far, and it looks like having been a good investment. Now, four factors at least played an important role when we took together the decision to create the IOB. First, there is a weak innovation in the area of eye care. Second, there is a high global medical need. Third, there is a proven advantage of collaborative scientific setup. And last but not least, the availability of highly talented experts here in Basel was one of the major reasons to get started. Now, the level of medical innovation in the area of ophthalmology has been low for many years, even as an increasingly high number of people around the world are suffering from eye diseases. Myopia is an example many of you can possibly relate to, which is affecting a growing number of people all over the world, especially in Asia. But the reasons as to why so many adolescents in particular are suffering from nearsightedness today are not yet fully understood, nor do we have adequate treatment options for many of the conditions which come as a result of myopia. We believe that this dearth of innovation is partly due to the fact that basic research and clinical practice do not collaborate and interact efficiently. There is actually no doubt that basic research is essential to the development of drugs. But a crucial element that drives the creation of new medicines is a real-world understanding of patient needs and a good grasp of the clinical practice. In the area of ophthalmology, we felt there was a disconnect on a global scale, and we wanted to bridge this divide by joining forces with the University of Basel and the University Hospital here in Basel. Both the university and the hospital had developed a strong clinical practice over the years, while at the same time, our basic research activities at the Friedrich Mischer Institute had gained considerable traction too. In many ways, this was a rare constellation. The more so because Botond Roska from the FMI and Hendrik Scholl from the University of Basel were two high-caliber experts who, we believed, could create unprecedented synergies by joining forces and sharing their experiences and insights under the umbrella of a new institute. As you will learn during the lecture, we believe the initial assessment was correct and the creation of the IOB was the right thing to do. As you all know, scientific progress will greatly benefit from improved collaboration across industries and research fields. We need to work with the best possible partners who have complementary knowledge and skills to bring us to the next level of discovery. At Novartis, we are engaged in hundreds of research partnerships all over the world, ranging across a diverse array of diseases and covering a broad range of different industries, including data and digital leaders such as Microsoft. In many areas, loose tie-ups with universities that have a single area of focus are sufficient. In other instances, we are engaged in longer-term deals with larger corporations to achieve lasting results. And as in the case of the IOB, The creation of a center of excellence is another way to go. While collaboration is paramount, there is, as we all know, no panacea for innovation and scientific discovery. As we at Novartis spend more than 9 billion francs on R&D each year, we must make sure we reserve our resources for projects that have the highest chances of success. In our complex and fast-moving world, this is not a trivial undertaking, especially if we consider that development times for a single drug can last a decade or longer. Making an investment in R&D not only needs expertise and the will to collaborate, it also takes courage and trust. Courage to bet on something that will be 10 years in the making, and trust in the ability of our researchers that they can find a way to develop breakthrough medicines that can really change the lives of patients. The researchers at the IOB, like their colleagues at Novartis, have what it takes to achieve this goal. They are leading experts in their fields and are willing to cross the boundaries of their respective domain to learn from each other and thus create new insights that can pave the way for cutting-edge therapies. Also, there is no guarantee that such a collaborative scientific setup will create breakthrough innovations. It is the most suitable environment that lends itself to generating discoveries that can change the practice of medicines. And I'm resting assured that our colleagues at IOB will succeed in this quest. That's actually all I wanted to say, and now I hand over to Hendrik and Botond. Please.
Yeah, dear Dr. Reinhardt, dear colleagues, dear guests, Professor Roska and myself would like to thank very much for this invitation. It's an honor and pleasure for the two of us to discuss today a topic that is obviously close to Botond's and my heart, and to present what we have achieved and what we aspire to achieve at IOB. I would like to start by showing you how much vision is being valued. When in the United States, in a national survey, more than 2,000 people unaffected by disease were asked, what is the worst condition that could happen to you? Blindness was rated very high. You can see that here. So it was rated as high as Alzheimer's disease or cancer, and clearly higher than HIV, loss of a limb, heart disease, arthritis, or deafness. The picture becomes even clearer when we look at the response to the question, what is the condition that you would feel would have the greatest effect on your day-to-day life? Here you see that losing eyesight is by far the most impactful condition, followed by losing memory, losing a limb, losing speech, or losing hearing. At the same time, and it was already mentioned by Dr. Reinhardt, eye conditions are on the rise. Here's data published in Lancet, where we see global trends of either blindness, defined by the World Health Organization here in blue, or here in red, that refers to people that are moderately to severely vision impaired. And then from the year 2020, that's the dotted line, that is the projection of people that will, in the next 3 decades, be affected by either moderate to severe vision impairment or true blindness, that is shown here in the blue line. So eye conditions are on the rise. However, to develop therapy that is still lacking, there are specific opportunities that we have in the ophthalmic field. And they are threefold, and I will walk you through all those 3 conditions. One is the optics to image the eye. The second is the immune privilege of the eye. And the third is the surgical accessibility of the eye. So please bear with me for a moment, a little bit of anatomy. Since I'm a professor in medicine, we have always start with anatomy. So that's a section through the eye. Light would enter here from the left, through the cornea, through the anterior chamber, through the lens, through the vitreous body that fills the eye, so to speak. And then light would hit this delicate structure over here in red. That's the human retina. So this is a 200 to 300 micrometer thick, transparent structure. But it has a very delicate structure, shown here on the right in high magnification. So it's layered, and Botond Roska will walk you through later in more detail. We see the photoreceptors, the bipolar cells, and the ganglion cells. Clinically, in clinical routine, we look onto the retina. This is how a human retina would look like. So we see this orange, reddish background coming from the layer underneath the retina. We see the optic nerve head where all the fibers leave the eye and travel to the brain. We see all these retinal vessels over here that nourish the retina. And then what we also see, at least indirectly, is the very center of the retina, which is called the macula, the human macula. And here we see the human macula in histology. This is how the human macula would look like ex vivo under the microscope. What we see is this so-called optical pit here. So we see that the secondary cells are displaced to the side to allow the very center have a high concentration of photoreceptors and to allow the human eye to have very high visual resolution. And now in a second I show you what happened in imaging of the eye, really a revolution over the last couple of years that has happened, where in a couple of seconds in a clinical setting in vivo we can obtain an image like that is about 100 times higher resolved, higher resolution than the best MRI or computer tomography on the market, simply because we have to go through optical transparent structures in the eye. We don't have to go through bone as an example. What you see is, again, this highly organized structure with the photoreceptors, the bipolar cells, and the ganglion cells. So this is something that we now use in clinical routine, and at IOB we go beyond, and we also can do post-image processing. And this is an example here. This was developed by the ophthalmic imaging group at IOB. And this is an example on a choroidal tumor. The tumor is shown here. This is this grayish structure, oval structure at the back of the eye. So we see here an infrared reflectance image. This is the optic nerve head. So now what we do is we image this structure here in a 3D volume scan. Then we apply a speckle noise removal to make the image much sharper and much crisper. And now with a technology that we call segmentation, we segment out all the structure that does not belong to the tumor, and then at point number 4, we arrive at the tumor. And this is, you can call it, with image analysis, a tumor biopsy that we can achieve by applying high-resolution imaging plus post-image processing. What we also can use is adaptive optics imaging. That allows us to correct for aberration in the eye and now look onto the retina, and what we then see in vivo are individual cells, and this is the layer of the seeing cells, the photoreceptors, the holy grail of vision. This is a resolution that is unprecedented by any other field in medicine, and this is a specific opportunity we have in ophthalmology. The second one is the immune privilege of the eye. The retina is protected in 2 ways. It's protected by the inner blood-retina barrier, so which means that any immune cells cannot easily travel to the retina when they arrive through the vessels in the retina. And the second one is the outer blood-retina barrier that protects the retina from the outside. And this is a specific opportunity we have because it allows to access the retina and apply specific therapeutic intervention with only limited inflammation. And the third is surgical accessibility. And this is a real-time video. So this is what we do every day. So, this is the so-called 3-port pars plana vitrectomy. So this is the first port. The first port here, we see now that the infusion line goes into the eye, because when we operate, we need to have constant pressure in the eye, right? So this is port number 1. Now we insert port number 2. Port number 2 is the main port for the retinal surgeon, because to the second port here, we apply the main surgical instruments in the eye. And then we place a third port. The third port is important because what we do during surgery, we have to illuminate the eye from inside. Keep in mind, and that may be a bit counterintuitive, in the operating theater, it is dark. The only thing that is lit up is the retina. And this is shown here. So now we have a surgical field here. This is about 3 millimeters, so it's a very delicate surgery here. Everything is dark in the operating theater. The surgeon has in his left hand the light pipe illuminating the very center of the retina. And here you can see that with specific instruments, a little membrane, only 10 micrometers thick, which is 1% of a millimeter, can be peeled away from the foveal center of the macula. So surgical accessibility, allowing such delicate procedures, is the third major opportunity we have to develop therapy. I think it's a fair statement to say that gene therapy has now arrived in the eye, and there are specific opportunities we have, but gene therapy is the major one. Those therapeutic opportunities are shown here. So on the x-axis, with time in this direction, we see progressive photoreceptor degeneration. If the seeing cells are still there, we can apply gene therapy or pharmacotherapy. If the target cells have died away, then we have to use other approaches like optogenetics or stem cell therapy. And in our lecture today, we will concentrate on the 2 main approaches here, gene therapy and optogenetics. It's not coincidence that gene therapy is very prominent in the retina field. The human retina is the tissue that is most affected by inherited disease. This is the revolution over time that we have witnessed over the last decades. It started in about the 80s or so, where the first genes were cloned that, if mutated, give rise to a retinal disease. And over the next decades, researchers have found many more genes that, if mutated, lead to retinal degenerative disease. To date, we know of more than 270 genes that, if mutated, give rise to retinal degeneration. So it's not coincidence that when we look at the list of approved therapies by the FDA, approved therapies for gene and cell therapy, that the retina, the human eye, is one of them. And this is the therapy that was approved in December 2017 in the United States by the FDA. A couple of months later, I had the honor to be the medical expert to meet with the European Medicines Agency. The European Medicines Agency in September 2018 still met in London, as you all know, because of the pending Brexit. The EMA has now moved from London to Amsterdam.
You have heard that about the value of vision, about blindness, and about a new therapeutic modality, gene therapy, to treat blinding diseases. In order, however, to develop effective gene therapies, we need to understand the functional architecture of the visual system, the hardware for human vision. So vision starts with a movie that enters your eyes. It is captured by the retina, and then it is broadcasted towards the center of the brain, at the lateral geniculate nucleus, and then arrives to the primary visual cortex. From here it radiates to a large number of other cortical areas and meets with expectations and memories. In your brain, in the human brain, around 50% of the cortex is devoted to vision. Although the retina is a tiny part of the visual system, almost all human visual diseases are localized to the retina. That gives its importance for research. The retina is not only a camera, but it's a biological computer. There is one video that pours into your eye. There is around 30 retinal movies that leaves the eye via the optic nerve toward the brain. Each of these movies are pulling out a different description from the visual scene, a different feature. So you get a 30 very highly compact representation, and then your brain is actually watching these retinal movies. It does not have access to the visual scene, but it's concluding what's out there based on these movies. If you look into the retina, what you see is a zoo of different morphologies. Each of these different cells has different shapes. And the key observation from Ramon y Cajal, a famous anatomist, was that indeed it's not just a chaos of many morphologies, but every morphology, you can find it many times. It repeats itself, that give rise to the idea of a cell type. Like this would be a cell type. The first cell type in the retina are photoreceptors, and you have two kinds of photoreceptors, the rods and the cones. The cones are for daily vision, shown here, and a single cone is watching one pixel on Federer's face and recording the light intensity there. And then it's talking to a next cell, a next neuron called the bipolar cell, and bipolar cells, as any other neurons, are similar to a phone. It has a receiver, it listens to the cone, and it has a transmitter, it talks to a next cell. However, unlike regular phones, the bipolar cells have internal computation. They distort the information, they modulate this information. There's not only one bipolar cell that listens to the cones, but many different ones, and each of them modulates in different ways the message that the cone conveys. Therefore, the visual information now joins parallel pathways. The next cell in the circuitry is this red cell called the emigrant cell, and this is fundamentally different from bipolar cells. Bipolar cells, as many other neurons in the nervous system, are excitatory. That means when they talk to someone, they activate that person. They increase the urgency of that person to talk again. The emigrant cell, however, is the opposite. When it talks to a cell, it inhibits, it vetoes that person to talk. Now, the final cells in the retina is a ganglion cell that listens to the bipolar cells and emigrant cells simultaneously. It's always on a conference call. And when the bipolar cell is alone saying to talk to the ganglion cell will send a message to the brain in a form of a spike. However, when the emigrant cell speaks at the same time as a bipolar cell, this message is vetoed. Therefore, what we see here, the retinal circuitry is an input-output machine. There is an input to the photoreceptor, the changing light intensity at that pixel, and the output are the spikes, these messages that the ganglion cell is sending to the brain, and you know every time there was a spike, the bipolar cell alone spoke, and when there is nothing going, either the bipolar cell was not active, or the emigrant cell vetoed the bipolar cell's activity. Now, we can simplify this drawing into a more conceptual representation shown here, that instead of these morphologies, we show units, numbered, these are the different cell types, that are connected together. And this such unit, this little circuitry, is watching one pixel on Federer's face, but as many of you know, Federer has many pixels, and therefore our retina evolved, put these circuitries all over the retina, and these little circuits talking to each other are not entirely separated. This circuit mosaic is now acquiring an image via the photoreceptor mosaic, and the ganglion cells now pulling out information, and then this feature that is shown here, and transmitting towards the brain. Now, what you can see here, there are three kinds of computation. There's a computation within the nerve cell, that's the telephone that modulates. There's a computation within the circuitry, the conference call, and there is also computation across these circuits on a mosaic. Now, your eye and every retina across vertebrates has 30 such mosaics squeezed into the retina, and therefore you have 30 movies, different movies, that are extracted from the visual scene and that is broadcasted to the rest of the brain. Now there are two components of what I described which is relevant for human disease. The first component is the computation, since if the computation is distorted, this retinal movie is distorted, our brain will have a distorted percept because it does not have access to the visual scene. So what it will conclude about the visual scene will be simply wrong. The other important component is that the hardware is very important to understand where disease genes, these 271 that Hendrik talked about, which cell type they are localized. But how do we figure this out in a human retina? Regarding computation, how do we measure computation by a human retina? And indeed, we need two components. One component is a chip, which it's made at ETH in Basel by Andreas Hirlemann's lab. It has 30,000 electrodes. We can put a retina on it and can measure for ganglion cells. We can actually measure the message that normally a ganglion cell would send to the brain, since we can project images to this chip. But a second one, which is very important, we need a human retina that is responding to light. And over the last years in IOB, we worked out a procedure together, the clinicians, the researchers, that for the multi-organ donor, within 5 minutes, we can take the retina in an oxygenated solution, we can put it on his chip, and after many, many trials now, we finally can record from thousands of ganglion cells, human ganglion cells, simultaneously the message that it normally sends to the brain. But you see here, we are flickering the light, and you can see around 1,000 ganglion cells responding real time. These are human ganglion cells. We can record for more than 12 hours, and therefore we can determine precisely what are the 30 different representations that sent to the brain by humans. The second is about the hardware. How do we determine gene expression in human retinal cell types? Now, we have in hand a human retina from these post-mortem donations, which we know are healthy because they're responding to light, and we can dissociate them to single cells, and then from each cell, using microfluidic technology, we can record gene expression. We have around 100 or 200,000 cells from which then we create a map. This is a gene expression map on which every dot is representing a cell, and 2 dots are closed if the gene expression in the 2 cells are closed. And what you see here on these maps are these clouds in different color representing the cell types of the retina because they have similar gene expression pattern. So what we can do now is take the average gene expression pattern, and now we have a map. We have for every human cell type, we have a gene expression pattern. And what we learn from it is that the cell types of the human retina have a unique gene expression. That means at least there are 20, 30 genes in each of the cell type that are uniquely expressed. Once we have this gene expression map, now we can proceed to the most important step. We can take disease genes that are sequenced or determined by human geneticists and map them to the cell types in the human retina where they express. And this is the foundation for therapy. So we get these maps. Indeed, these maps are much longer. I just show you partially. Each row is a disease gene that we obtained from colleagues in human genetics. And each row is a cell type in the human retina. And although I don't want to focus on the details here, there's one important message that it's clearly visible, I think, for most of you, that the human diseases are cell type specific. They are not all over expressed, but only particular cell types. For example, this first gene is expressed only in these cones, or then this one only in rods, and it can go on and on. And because diseases are cell type specific, the concept of cell type is not only the basis for understanding retinal function, but also the basis for developing therapy.
So before we get to specific therapeutic approaches, I would like to introduce a condition with the name retinitis pigmentosa. So in order to understand those conditions, I would like to show this human photoreceptor topography map. So this is essentially the density of the seeing cells in the human retina. We see on the left the cone density, the cone photoreceptors, and we see on the right the rod photoreceptors, or the rods. Cones are active under daylight conditions. They allow us to have fine detail vision and allow us to see colors. Rods that are, by the way, 20 times as numerous than cones allow us to see under dim light conditions. Conditions that exclusively affect the cones and the macula would lead to symptoms right in the very center and would spare the rest, while conditions that affect rods and secondarily cones would first affect the mid-periphery and then later also the center and also the far periphery. So retinitis pigmentosa, what is that? Retinitis pigmentosa is a term given to a set of inherited retinal degenerations that feature the degeneration of rod and cone photoreceptors. The worldwide prevalence of the condition is about 1 in 4,000 individuals. That makes up for a total of more than 1 million affected individuals worldwide. When we study the impact of vision by the disease, that typically there is some remaining periphery and some remaining in the center, and then later the periphery can also be lost and people develop something that we call tunnel vision. What happens in retinitis pigmentosa? What we see here is in this column is a normal human retina. Here a sketch of the human retina. And on the right we see an affected retina by retinitis pigmentosa. And what we see is that the outer segments of the photoreceptors that make up for the light sensitivity of the seeing cells of the photoreceptors are lost, while the inner retina is still in relatively good shape. Again, I show you this normal fundus because I would like to contrast that with an affected retina. I may actually disclose that I'm specifically happy about that normal fundus. I will now enlarge this part, so it's my retina. When we look at the fundus, the retina of an affected individual, we see these pigmentary changes here in the mid-periphery that give the disease its name. So this is pigment migration into the retina that always comes together with degeneration, which means that a patient like this one would not see around here, but may retain vision in the center. We also see that the retinal vessels are attenuated and the optic nerve appears a bit pale. In order to visualize for you the symptoms that patients may have, I would like to show this video. So just imagine a scene in the Brussels underground. So here now we try to mimic what a patient may see with retinitis pigmentosa. There is some problem with the contrast sensitivity, so the contrast is not as crisp as in a normal subject. And the subject has lost mid-peripheral vision. So the subject needs to scan things in order to find things. For example, here going to this shop or find a specific book means the subject needs to scan and use this remaining island of vision that he or she can use. And now mobility is also an issue, now getting to the counter. The problem is that with the condition, the visual field is restricting itself more and more, and also contrast sensitivity is reduced more and more, and eventually, not for all patients, but at least for some patients, the light will go off completely, turning those patients in complete blindness.
If you look into the retina of these completely blind patients with retinitis pigmentosa, what you see that the rod photoreceptors are all dead. The cone photoreceptors lost their light-sensitive compartment, the outer segment. Sometimes their cell body is still intact, but most of the other cells in the retina are normal. That means that the computer is there, but the camera is lost. And the therapeutic idea that we have in IOB is to sensitize the remaining retinal cells to light using genetically encoded light sensors. This is called optogenetic therapy. And the simplest and most robust light sensors are made by algae or bacteria. So that's where we obtain these genes. We then can come up with different strategies, which cell type to sensitize to light. We can sensitize the remaining cone cell bodies, the bipolar cells, the so-called A2 emigrant cells of the ganglion cells. As you see that if you sensitize the cones, the remaining circuitry will work for you. If you sensitize the ganglion cells, the circuitry of the retina will not be active. So therefore, our focus in IOB is to resensitize the cones, the remaining cones, to light when it's possible. Now, how do we target this light sensor made by algae to one of the cell types of the retina? What we do here is use light sensor, so we use adeno-associated viral vectors, which are shown here. These are little balls. This is called the capsid. It's a protein ball. And within the protein ball, you can see a single-stranded DNA that has two important parts. One is the gene we want to deliver. This is the light sensor. And the second one we call a promoter. The virus will enter all cells of the retina when Hendrik injects them into the retina, but then only expresses a light sensor in the ones we wanted because of the presence of this magical word promoter. This is indeed a DNA sequence. So the question, how can we find this DNA sequence? And the idea is relatively simple. We know that a specific cell type in the retina can express a specific gene because it has an internal promoter. So therefore, we look at this gene expression pattern I talked about and trying to learn which are the sequences that are necessary for a cell to express a specific gene, and then we make educated guesses and make synthetic promoters and test them. So recently, we made around 300 different AAVs with different promoter sequences, guessed sequences, and then, of course, we have to test them, and the best to test them in the human retina, because that's what we want to treat. And then here comes an important technology which was developed by our collaborators and friends, that Arnold Szabo and Serge Picot, that after multi-organ donation, using their technology, we can keep the human retina alive for 14 weeks. The human retina is enormous. It's like 70 times of a mouse retina. We can cut it into small pieces, and every piece we can drop one of our viruses to ask which cell type, if at all, they express the sensor. And this project was indeed very highly successful because after several years, now we have in hand several promoters that specifically target retinal cell types. For example, ProA7 promoter, this is a side view of the retina, expresses here this green fluorescent protein only in cone photoreceptors. Here's the top view, what you see. And we know they are cones because we have a marker that co-express in the same cells. We also have promoters for rods, also not as good as cones, as well as other cell types. And we have a large list now of promoters can target different retinal cell types. Now for optogenetic therapy, we choose the one that target cones, shown here, express the light sensors, and ask if they can resuscitate this human retina in culture. And remarkably, 9 weeks post-mortem in a culture, once we express an optogenic sensor in cones, the cones start to respond to light. Here is showing light pulsed at different intensities. This intensity, you see cones are responding. Then cones are talking to the bipolar cells. Bipolar cells also start to respond. Bipolar cells talk to the ganglion cell, and they also respond. So 9 weeks post-mortem, we have a fully functional human retina in hand that is telling us that this approach might be a one that we will choose going ahead for clinical trials. For clinical trials or for clinical use, we need something more, namely these bacterial light sensors have a specific wavelength they require, and they cannot adapt to all the intensities that you can do. They have only a narrow range. And therefore, our collaborators at GenSight Biologics, they made a goggle that has a video camera looking outward, watching the scene, and then a projector that projects a particular wavelength to the retina, and in between the computer that we can add some computation. And this particular wavelength will then excite the artificial photoreceptors to provide, we hope, some form of form vision for our patients.
The second condition that we would like to introduce is macular degeneration. Age-related macular degeneration is the leading cause of blindness in the industrialized world. And here we see a video that shows how this looks like. We look at the macula here. We see those drusen develop. We see also some bleeding and some scarring. It all affects the center of the retina, namely the macula. And here we see the consequence for vision. It means that in the center, the vision becomes blurry, but patients retain the peripheral vision. I would like to show again the human photoreceptor topography and again point towards the left where we see the human macula and we see this high density of cones. And this high density of cones is then being affected by the disease and then causes specific symptoms. So what is Stargardt disease? Stargardt disease is the most common juvenile macular degeneration. Juvenile meaning it starts earlier than age-related macular degeneration. It shows a prevalence of 1 affected in 8 to 10,000 people. And the underlying gene, as a disease gene, accounts for about 7% of all inherited retinal degenerations. It shows autosomal recessive inheritance. That means that parents typically are unaffected. They carry a mutation. Carrying one mutation would be compatible with a completely normal retina, no disease. But 1 out of 4 children, statistically, would inherit 2 mutations at the same time. And in this scenario, the subject would be affected. The underlying gene is called ABCA4. It's a so-called ABC transporter. And a gene defect in ABCA4 leads to abnormal metabolism in photoreceptors. So it is shown here on the left. These are the outer segments of the photoreceptors, either the rods or the cones. We have these little discs here. On the left, we have a healthy ABCA4 and a normal metabolism. And on the right, we have a disrupted ABCA4, and therefore we have also a disrupted visual cycle. That means that the visual pigment cannot be restored completely or not sufficiently. And as a side product, we see accumulation of toxic material in the retina. Unfortunately, the gene is very complicated. It's a large gene. To date, about 1,000 mutations have been described. In our own study, we discovered 50 novel mutations alone that give rise to the condition. Fortunately, not all mutations show the same prevalence. There are some mutations that are specifically prevalent. One of them that we aim to target shows a prevalence of more than 15% of all mutations that we see in Stargardt disease. Clinically, when we look at the retina, we see the right eye here, the left eye here. We see the optic nerve and the vessels. We see those flecks that were described by Dr. Carl Stargardt exactly 110 years ago. And those flecks already indicate this toxic material that accumulates underneath the retina. And this can be even better imaged by a technology called autofluorescence imaging. Here we see this material lighting up as these whitish dots. We also, with this technology, see the consequence of the toxic material, and that consequence is now imaged in the very center of the retina, in the foveal center, where we see gray, dark gray, or even black areas, and those indicate that the healthy signal that shows this mid-gray level is being lost, and this correlates with degeneration in the outer retina. This can be tracked over time. So this is a video of a patient of mine that was imaged every 6 months. And over those period of 2 years, we see that the central lesion increases over time. Yeah, you see that here it gets larger and larger over a period of 2 years. We developed that further. We defined those lesions and then made them a real outcome measure that can be used in a so-called reading center. And the FDA accepted this outcome measure as a primary outcome measure for clinical trials in Stargardt. Not surprisingly, because what we like to preserve is vision, and those lesions are tightly correlated with visual loss. In order to measure visual loss, we use a technology called microperimetry. Here you see a patient sitting there. The machine projects a stimulus to the retina. It can also correct for eye movements. So every 20 milliseconds, the machine corrects for any eye movement that can happen to precisely stimulate a specific site at the retina. And then we can superimpose a functional map onto the retina. So we remember that patient here on the right, we see this lesion here, and now we superimpose a functional map onto the retina. Everything that is color-coded in red means the vision is severely compromised, and everything that is color-coded in green, just around that lesion, means that sensitivity is relatively preserved. What we also see, by the way, is that that patient lost the very center and now is using an area outside the very center. So the patient also knows that she or he needs to look to the side to see relatively well. Unfortunately, such an area here has not even close of a visual acuity than the original very center that we have in the retina called the foveal center. Eventually, what we would like to do, and what we're already doing, is to have a tight correlation between structure and function. So here you again see an infrared reflectance image. This is this high-resolution OCT imaging where we see all these retinal layers. Now we superimpose a functional map onto the retina. Every little dot corresponds to a measured threshold, so it shows how well that subject is able to see. And we can do that in close correlation with other modalities. Now we switch to another modality of imaging, and again we can go point by point and perform structure-function correlation.
As you heard, there are a large number of mutations that leading to Stargardt disease in the ABCA4 gene, but one of them is highly frequent, and this is where we concentrate in our therapy development. This is a GGA to a GAA change in the genome, such that ABCA4 instead of a glycine now codes for glutamate that compromises its function. Now, the therapeutic idea is rather simple: to change a single base of the 3.2 billion bases of the human genome to restore the normal ABCA4 sequence. In order to you to experience the challenge, you could go to the Wellcome collection in London, where you see a shelf where the entire human genome is written in books, in 100 books, organized by chromosomes. And to find the particular mutation, you have to take one of the books of chromosome 1 and then find the particular sequence you're looking for, take an eraser, and then write the right character here. Of course, the characters here are only 4 of them. You can find 4 of them because the genome has, instead of the English alphabet of 26, had only 4 characters, A, G, T, C. Now, molecular engineers had an idea for a repair tool, what you just heard, which is similar to a track repair machine, a train track repair machine. The train tracks, some of the ties sometimes are faulty, and therefore there is a machine that goes there, finds the faulty location, it has some information, and then has some tools to repair the tie. Now, the molecular equivalent of that, of the train track, is a DNA, and the ties are the base pairs, the AT, for example, and you can see that this is a faulty pair in the case of the Stargardt disease mutant, and this is now an enzyme, a molecular machine that has 2 important capabilities. The first capability that molecular engineers, they put a sequence on it that serves as a guide to find the sequence in the genome and bind to this location. So now you already know where is the fault, and then it unpacks an enzyme that binds to this A, the faulty A, and converts back to G. Now, of course, we need test system to test in a laboratory how well these base editors are working. The first test system we are using is in cell culture, made by IOB engineers, and we call it a traffic light reporter. It's traffic light reporter because we engineered the genome of these cells to express potentially two different colored fluorescent proteins, TD tomato and GFP, but this ABCO4 sequence, the mutated, is put in between these two genes, and that the GAA is preceded by a T such that implementing a stop sequence. So when the cell would like to translate this across these two proteins, it will, to RNAs, it will stop at this point, it fall off, and therefore GFP will not be expressed. It only can make red fluorescent proteins. However, if the base editor corrects A to G, this is a read-through signal, so you can make both a red and a green fluorescent protein. So we test it in cell culture. You can see that when we have the disease mutation, then you only make a red fluorescent protein. However, when the base editor is added, now most of the cells are converted also to green, suggesting that the base editor efficiently editing the ABCA4 site that we ought to edit in patients. Of course, these are cells, human cell lines, which are not photoreceptors. We have to, before going to the clinics, we have to test base editing in human photoreceptors in their normal environment in the retina. The dilemma is that it's very unlikely that we get a postmortem donation from a patient with Stargardt disease. It's very rare. Therefore, we need a technology to build Stargardt retinas in a dish. And this is what I described to you. We start with a skin biopsy from any humans, indeed from any age. And from this skin biopsy, we take fibroblasts shown here and convert them to induce pluripotent stem cells using Yamanaka factors. And then, using a technology that originally developed in Japan, we can induce with small molecules the formation of optic cups from this cell mass. These are human optic cups that we can remove and start to culture them. And around 80 days, what you see that these tissues start to polarize. In one side, the photoreceptor precursors are forming. On the other side, the ganglion cell precursors are forming. And it took us a long time to figure out how from this cell mass we can indeed make a real 5-layer tumor retina that Hendrik showed you before. But we have some really good engineers in IOB that figured out after a while that at the beginning, we have to work on the IPS clones, and if we get the right clone, then we can develop within 200 days a human retina in a dish that has now all the 5 layers, photoreceptor layer, the bipolar cell layer, ganglion cell layer, and the synaptic layer in between. And comparison, you can see here a human retina. These organoids not only have the layers, but they also have the outer segments, the light capturing elements of the retina. And if we take sections one by one, we can see many of the famous cell types of the retina. For example, the human cones are forming in these organoids, just like in the human retina. The rods are also forming. Not only the photoreceptors are there, but the photoreceptor for color are also forming. You can see the blue cones and the red-green cones. If you use other markers, you can see the bipolar cells, the horizontal cells, the Muller cells, and we can go on and on to show that almost all cell types are forming in a dish that you can find in a normal human retina. Moreover, you can see connection between these cells. They are talking to each other. These are so-called ribbon synapses, where the cones and the bipolar cells are talking to each other. But can Stargardt disease be modeled with retinal organoids? Of course, what you have to look at is gene expression in the different cell type of organoids and compare them to the normal retina. And the way we do it, again, dissociate the organoids to single cells, use single cell sequencing, and put the disease gene map of the normal human retina and the organoid side by side. And if you look at it, here you can see the adult human retina, the retinal organoids, and if you look at it, they are rather similar. But is every detail similar? No, they are around 70% similar. But however, we get lucky when we look at the ABCA4, when we zoom into this map, look at ABCA4, in the peripheral retina, you can see expressed in the photoreceptors, and you see a similar expression in organoid. So we can study this disease. So the next step is to make an organoid expressing the GAA mutation that's already made in IOB. And finally, we need now AV vectors that can target the relevant cell types, in this case the cones, and we have an AV with the right promoter that can target almost 100% and exclusively the cones in retinal organoids. So now in the next month, this is where we are. Now we try to bring in the base editor to cones, and hopefully in the next months we are learning whether how efficiently we can engineer and edit the right bases in cones, and we hope in the next year, if this is working in organoids, we can bring it to clinical trials at IOB and the eye clinic. At this point, I like to summarize our talk. I started telling you that vision is important for humans. It starts in the retina, and then it is broadcasted towards the cortex, and around half of our cortex is devoted to vision. And despite the retina all being a small part, almost all visual diseases are localized to the retina. And indeed, it's very lucky that's where these diseases localize, because as Hendrik told you, the retina is an accessible part of the brain, and they have quite a privileged immune status. Nevertheless, the retina is not a simple camera, but it's a biological computer that is extracting different features from the visual scene. And if we zoom into the retina, we see a lot of different morphologies that are grouped together, so-called cell types. And cell types are the basis for understanding retinal function, to understanding disease mechanism, and to develop therapy. In order for developing therapy, we need human models. And I showed you two human models. The first one is a postmortem retina that can be kept alive for 14 weeks. And here we can study function. We can look at gene expression in the human retina, map diseases, and different vectors, develop vectors to target cell types. The next model system is retinal organoids, where we can study disease mechanism because we can engineer these organoids to express their genome. We can engineer. We can also take them from patients. And finally, we can develop therapies in organoids that later can be brought to clinical use.
So, ladies and gentlemen, I think it's a valid statement to say that gene therapy has entered medicine as a third therapeutic modality. We entered a new era of medicine that you see here on the right, and gene therapy allows us to use DNA or RNA. It may, at least in some instances, allow to perform a one-time treatment, and the effect of the treatment may be permanent after a single administration. And it may be potentially curative. That means that it may allow to halt the progression of a disease or to allow alleviating the underlying cause of a disease. My statement would be gene therapy naturally fits the eye. I would like to conclude with a video here. So again, we are in the eye. The light pipe comes from the left. Therefore, you see a shadow of this small cannula. It's a 41 gauge cannula. Again, this is 3 millimeters here. The closer the shadow gets to the retina, the closer we are with the cannula. So the cannula will, in a second or so, penetrate the retina. And now, in this moment, we can deliver the gene therapy product underneath the retina with all its implications, immune privileged, closed, and gets exactly where it needs to go. I would like to conclude here that eye therapy will be in the center of medicine. And it's also important that Botond and I really value the contribution from all of our IOB members that are shown here that provided the material that we have shown you tonight. I would like to conclude here by showing the team and thank youing the team here that came together at IOB's first retreat in Vevey at Lake Geneva. And you see here almost 80 members of IOB. And keep in mind, this was just 1 year after IOB became operational. We would like to take this opportunity to also thank very much the founders, Novartis, Basel University, and University Hospital Basel for having made all this possible, and Basel-Stadt for its generous support of IOB. I would like to thank you for your attention tonight, and Botond and I are very happy to take questions. Thank you.
Okay, I have to say, wow. Thank you very much, Botond, very much. Thank you, Hendrik. I mean, I had a steep learning curve. I guess some of you too, I hope at least. We're going to do Q&A now, 15-20 minutes, as long as it takes. If it takes longer, we can continue at the apéro. The two gentlemen will be with us for the apéro. 3 little remarks for the Q&A. You can ask questions in German or in English. We can handle both. Second remark, please wait for the micro. So there are mostly ladies in the corner giving you the micro, so before you speak, please wait. And the third one is, these are 2 medical doctors, but please refrain from asking questions about specific conditions that you might have or your relatives might have. This is not the place for that. That is a remark out of experience. And since I'm standing here, I'm just curious, and sorry, I just can't hold back, just one little question. When the video from Roger Federer and this optogenetic vision restoration, I mean, what do people see after that? I mean, if we ever succeed in doing that, what will it be? Can you describe that, or is that?
I think it depends on the approach. I showed you there are 4 different approaches, and the resolution will be very different. In each of the cases, we will see grayscale images. The best resolution will come with cones, and the worst probably with ganglion cells. Okay. We will never achieve probably the normal resolution of the human macula.
Okay. But still.
I think it's fair to say that such patients undergoing such type of therapy will undergo also a learning curve, hopefully a steep one. We have experience from other approaches like cochlear implants or retinal prosthesis, where indeed patients have to relearn to hear or to see, and we believe the same would apply here.
Okay, thank you very much. So now enough from our side. The floor is yours. So we have a question down here, 3rd row.
First, congratulations to your wonderful work and excellent presentations. I wanted to ask you, in the case of blindness, do we have to expect any structural or functional changes in the cortical area, which may hamper then a success you may have on the retinal side? Thank you.
I think that it depends on exactly which condition. I think when the optic nerve is intact, it's very unlikely, or when we look at patients, what we see is that functionally the hearing or other senses or even calculation can take over the brain, but there's no really major morphological changes. When the optic nerve is lost, then at the lateral geniculate nucleus, the second stage, there is a loss of certain cells called the magnocellular cells. But fortunately, those that are there for high-resolution vision, they are rather intact.
Yeah, maybe I can add that we see patients that lost significant vision, and then they are so-called filling in phenomena. So, I mean, the brain is still there, and the structure that would obtain information from the brain is still there. And there's a specific symptoms that patients report, that, for example, visual hallucinations, because the brain is still active and it fills in information where there is no information. So the question is, can this brain areas be repurposed for other purposes? This is not our field. The answer is most likely to a little degree, but not to a major degree, because it's so specialized.
Thank you for the outstanding presentation, also for including patient perceptions. I have two questions. The first one is, if you record the excitatory patterns of the ganglion cells, how much does the signal vary from person to person? And then the second is, if I understood correctly, you only introduced the glycine glutamate mutation once the organoids were fully differentiated. And is there any downsides to doing that while it's still in the iPSC state?
Yeah. So regarding the first question, that we cannot yet tell the variation from human to human, I think that we need much more samples. What we can tell, however, when we look at the single cell gene expression, is almost no variation across 3 subjects. So very, very minor. That was very surprising for us, telling us that the cell type composition at least is highly conserved. Now, regarding the use of retinal organoids as a model for adult retina, what I didn't show here, but we learned that the rate by these organoids develop are identical to the rate they develop in the, as a fetal retina in the mother's thumb. Therefore, a 36-week-old organoid would be a newborn. So I think this is really good to model genetic diseases, early genetic diseases, and not as good to model, for example, macular degeneration or age-related diseases.
Thank you.
So thank you very much for this excellent presentation. I would have one question, that what is the bottleneck in this research? So you mentioned that you need, for instance, post-mortem retinas, and do you have enough of those? Could we help by offering if I die on the bicycle home?
This is a very, very important point. The biggest bottleneck for us is to obtain post-mortem retinas. And any help from the community, even we now reach out to RAO, we try to reach out to our neighbors in Germany, but our biggest limitation of the IOB is a rare condition, the rare case we get in human retinas. We're collaborating also with, in Budapest, where they have a lot of 2 million, it's a large city, but the more we can get here in Basel, the more we can do for patients.
So up there, they may be faster.
Okay, whoever is faster wins. Yes, the lady in white.
Hi, yeah, thank you for the really interesting presentation. When you're operating directly on the patient, are you operating under a general anaesthetic or a local? And if it's local, how does the patient adapt to having that coming into their visual field?
Okay, I'm not sure if acoustically I got the question right.
Sorry.
Yeah.
So when you're operating, are you operating under a general anaesthetic or under a local? And if it's local, how do the patients adapt to seeing all the apparatus coming into their visual field?
Yeah, that may sound surprising, but most of the intraocular surgery can be done in local anesthesia. It doesn't have to be done in local anesthesia. It can be done in general anesthesia. The most important issue is that the patient may move. Most patients perfectly understand that they should not move while we operate. We typically then make agreements that the patient would, give a signal that he or she would need to move for a moment. So we would retract instruments out of the eye. But this sounds more complicated than it is. Typically, it's not a problem at all. But for some patients, we offer general anesthesia because the procedure from a surgical perspective is indeed safer when we do it in general anesthesia. But then you have to factor in the risk of general anesthesia. And there are always pros and cons. From a pure surgical perspective, I would always prefer a patient in general anesthesia. But in reality, I would say more than 90% that we operate are in local.
So the question I had was, towards the beginning of your talk, you mentioned that myopia is prevalent worldwide. So is there progress towards, let's say, reducing myopia? And what is the latest state-of-the-art research that is being done towards addressing myopia?
So the question was about myopia, progression of myopia, and so forth. So the projection that we currently have is that because myopia causes secondary problems, I think that that's now important to comment here. The secondary problems being myopic macular degeneration, retinal detachment, glaucoma, and cataract formation. And if we take them all together, the projection is that by the year 2050, myopia, not because of the refractive error, right? Not because of the refractive error, will be the leading cause of severe vision impairment and blindness in the industrialized countries. Okay. The research at IOB is not about those complications. We see those complications in clinic, and if there's a retinal detachment, we operate. If there's glaucoma, we treat glaucoma. And if there's myopic macular degeneration, we use other medications in order to address specifically this specific entity, because it's very similar to wet AMD. Okay, what we address at IOB is controlling eye length growth. Okay, so the problem of myopia is the eye is relatively too long, and therefore the visual plane is in front of the retina, and you can correct that with eyeglasses. Okay, there's a sensitive period that goes from something like 8-10 years to 16 years or so. It can continue to 25 years and even further. People with progressive myopia can develop more myopia even beyond. But the focus at IOB is, are we able to control eye growth? And we have specific technologies available that may allow us to be successful, because currently it's not understood how the retina actually detects the sign. For example, if the image is in front or behind the retina can detect that. It's unknown how that happens. If you cut the optic nerve, which you shouldn't do, of course, but the retina still detects if the sign is minus or plus, right? And the retina reacts immediately if the plane is not right at the retina. The choroid, which is the vessel structure behind the retina, will push the retina forward or behind. And this can be measured within an hour or so. Okay, more questions? Yes?
Yeah, thank you very much for the lecture. If at birth the cones are lost, for example, you talked about the computational power of ganglion cells and so on. How long is this computational power being held up, kept up, also in the cortex, so that your therapy is can be deployed after all? Thank you.
What is truly remarkable that when the photoreceptors have lost, you look at, Hendrik showed some human pictures, and we know that the ganglion cells and the bipolar cells are there essentially forever. There is never a condition, we say, that the bipolar cell and the ganglion cell lost. They might decrease by a factor of half, but they never lost. So I think that is a really, really good news, is that some reason, even though we have complete lack of input, the retina holds on. This is what we also see in animal models. We have animal models that are completely blind until late age. Then we reintroduce optogenetic sensors and very fine computations we can record in the ganglion cells. So that particular problem, fortunately, nature made the retina in a way that will not affect the outcome of therapies.
If I may add, there's a specific issue, and maybe you wanted to address that, is that there needs to be a visual experience in order to learn vision. That was the Nobel Prize. There are actually 3 Nobel Prizes in ophthalmology or vision research. And that was one of them, showing that if there is no visual input coming from one eye, you see that part of the cortex is not able to learn vision. Clinically, we call that amblyopia. So the question is, if there are no photoreceptors or no cones, you mentioned cones available from birth, right? Is it then later possible to relearn vision if and when we are able to resuscitate cones in the retina? And the answer is, we don't know. Nobody has done that yet. And it is a possibility that we may not get to what we are used to, namely 20/20 visual acuity. But I must say that bar maybe is already too high. And this is not the level of vision we want to resuscitate that we are talking about.
I have a question regarding the coverage of the costs. So it has been mentioned that these diseases are still rare disease, and there are still treatments explored or assessed in clinical trials. So who is covering the costs of these treatments, gene therapies?
So if it's a clinical trial, it will always be the sponsor who is covering the cost. So I'm not sure if you are getting at, for example, if the patient would need to pay for the treatment, the answer is always no. If it's a clinical trial, a patient would never pay her or himself for the therapy. It would always be the sponsor. I'm not sure if that answers your question, right? So until a specific treatment is not marketed, there's also no specific price tag for it. All the costs need to be covered by the company that is sponsoring a specific trial, and all treatment-related costs will be covered for the patient. There will typically be absolutely no cost for the patient.
And so, thank you. That was part of the question. And so, I mean, if there is a patient that is not participating in a clinical trial, so would there be no treatment or no coverage from the health insurance?
Okay, so this is now we get very much into regulatory issues here. So if there is a clinical trial and a patient would not participate, for example, the patient would not meet the inclusion criteria, and there are efficacy signals that were already recorded and published, there are specific possibilities called compassionate use programs where specific medications can be made available for such a patient. It's a very complicated regulatory process. There are typically agreements. For example, there's an agreement between France and the US, which is also the reason why the first gene therapy in the eye was performed in France. It was not performed because the medication was approved in France. It was a specific agreement between the government in the US and France that led to a compassionate use of the treatment in France. Okay? So this is just an example, but we get into very complicated regulatory issues here.
Okay. Next question, yes?
I would like to ask professors, first, my congratulations to you and your research work. I'm interested in the adult macular degeneration concerning subretinal extrude or not, wet and dry, and how is their progression and what it may be expected in the future, and do you predict to cure it or treat it by stem cells?
So that was a complex one. So let's break it down to so the question was regarding age-related macular degeneration. We have not covered it in our talk, and there are two late forms. One is so-called wet AMD, which in my opinion is nicely covered by the available medications on the market. Although there's still gene therapy being developed in order to continuously express compounds that would block VEGF in the eye to make those injections in the future obsolete. I'm not even sure if that's going to happen, but there are activities around that. So maybe this covers wet AMD. So when we get to late dry AMD, also called geographic atrophy, AMD is not a monogenic disease. We have two world experts at IOB that are dealing with genetics and also complex genetics. There are about 50 variants in the human genome that are associated with a risk to develop late AMD. A couple of them can be grouped to the innate immune system called complement. And there are activities to try to develop gene therapy to tackle the complement system in order to have a treatment effect on late dry AMD. There's a phase 1 clinical trial performed in the United Kingdom that specifically addresses that. The question further was, can we then use stem cell therapy to cure those conditions? Here, my answer would be, the current use of stem cells at IOB is not that we would use stem cell therapy to regrow the retina in a patient retina at the site. But we use stem cell therapy to grow the retina in the dish to perform experiments and develop treatments. In my personal opinion, that is a remote goal that we may be able in the future with stem cell technology to regrow the retina in a given patient at the site. Even if that's possible, to regrow the retina in its wiring that is specific for the fovea and the macula compared to the rest of the retina is such a high bar that currently I would believe that's an unrealistic goal.
Maybe I can add one point, is that I think one of the key questions is that what is causing age-related macular degeneration, and we do not understand the answer for that. So what in IOB we are focusing is try to understand the disease mechanism. And in my view, it's either one can get lucky with therapy, for example, but in the case we just find a molecule that cures a particular disease. But in IOB, what we try to do is first understand disease mechanism, and once we understand, then come up with treatment options. But right now, we do not understand how AMD develops.
Okay, so we have time for, sorry, just quickly, I'm a bit of housekeeper here, time for one complex question or two not so complex questions. Go ahead.
I had a question, but I guess it's related to the previous one. So what I understood from the talk today is that there is some genetic predisposition that is in some of us to develop eye disease. And actually, I guess, yeah, you kind of gave an answer that there is not a good understanding what role age plays in development of the eye disease, because if there is genetic predisposition, you might think that the disease could occur at a really early age, but for some of them, I guess it occurs later. And so, but I guess it's, yeah, I don't know if there is a understanding of why it's the case, and if you have any hints to what triggers the development of the disease, and what we or what patients could do to prevent the disease to develop.
Would you? I will just start maybe saying is that really there are two important groups. The monogenic diseases, we do understand what triggers, and in some cases we even understand why is it coming later. In a case of, it's an accumulation process of, for example, some toxic materials. In the case of polygenic or complex diseases like AMD, we truly don't understand how age contributes. That is unknown, and it's a very important question we and many others are after. But it's a difficult question because we have to wait a lot to age. So it will take a while.
Okay, final question.
So first of all, thank you very much for the great talk. And my question is also regarding the optogenetics approach. So instead of using an external device for filtering the light, have you thought about engineering the algae or the bacteria?
Maybe I can start, although really Botond is the expert. And the thing is that if we have a shift towards infrared light, we have actually an opportunity. It sounds like a problem that we would need to solve, but it's actually the opposite. Why? Because the light that enters the eye at that amount that we would need to stimulate artificially, right, is very high. If we would need to use blue light or normal white light like here, right, that could be harmful because of heat production and so forth. While using infrared light, we can more specifically penetrate tissues at a level that would be sustainable, right? But I hand over here to Botond.
Maybe to respond, the reason we need the goggles is that no matter what light source, a single gene can only mediate vision in 2 log units. And you can see across 10 log units of visual information, from 1 photon to 100 millions or more photons. So it's very important that no matter what we do with a single gene, it always be around 2 log units, and therefore we would always need a goggle. It can be more sensitive or less sensitive, but we will need a goggle.
Do we have a burning, short, non-complex question that we can still answer?
Keep in mind, I mean, we are the last bit here in front of the apero. So,
yes. You keep everyone from the apero, but no pressure.
You mentioned earlier there was this sensor that was developed. Do you think there's a possibility in the future of having some sort of bionic eye that assists patients who develop these diseases?
The goggle is a bionic eye. So what I mentioned, there is a computer interface between the video camera and the projector. And very important, in the IOB, we have several groups, including my group, who are working on mathematical models describing human retinal computations. And for example, if you go for bipolar cells, we implement a computation. We also go for cones, we implement a computation. It's not video camera to projection, but it's a computer. And so we do use bionic devices. It is an integral part of the optogenetic program.
Okay, so I think with that we close the Q&A. Thank you very much, Professor Scholl, Professor Roska. Big round of applause. Okay, before just those of you who want still to listen quickly for a minute, we have a next lecture already planned. This is about something completely different. It's not about science. It's actually about responsibility, social and environmental responsibility of companies such as ours. We have Professor George Serafheim, who is the expert on that field. We have him here next May. And he is asking the question, reimagining capitalism, big problems and big companies. I think it's going to be interesting. Hope you're going to join us there as well. And now enjoy Yapro. Thank you very much.
When IOB was founded, we saw 2 cultures, the medical doctors and the researchers. And 2 years IOB, what we see is a uniform culture when researchers talk to medical doctors and medical doctors are constantly talk to researchers.
So ophthalmic research, at IOB at least, takes advantage of key technologies that so far had not been available to anybody in the world. These technologies allow us, for example, to grow retinas in the dish and to perform experiments in a very short period of time that otherwise would need to go through various phases of development. We believe with such technology that we can bypass some development steps and arrive at therapy much earlier than in the years before.
In my opinion, we need novel approaches because we can then prevent diseases. We can, through, for example, gene therapy, we can very precise cure a person, and we go here towards personalized medicine, right?
I think that's a very interesting lecture, and because as they showed, there are a lot of blind people around the world, and it's increasing in the future. So that's a very hot topic to have doing science on. So I think it's very important that there is new approaches to find new treatments for all the blind people or all the people that don't see very well.
Yeah, I'm very much hopeful that me, my children, the next generation will sort of profit from what these people are doing.
What exciting times may be lying ahead in terms of being able to use new technologies, in terms of being able to use gene therapies, and obviously there is the huge need, and human vision is probably the most important thing that people have and value.
I think the approach with the organoids is very new for me and it's very interesting, so that you basically can rebuild a retina from scratch. That was very interesting because I think that's also it's very basic now, but if you think what can happen in the future with that approach, it's very interesting because you basically could rebuild the eye.
I would say the three big pillars of medical science or therapeutics would be, the traditional chemical molecules, biologics, and I think the most exciting pillar is gene therapy. So this lecture, and which is in collaboration with our organization, was showcasing what amazing advances in ophthalmology area are happening in gene therapy. And I think as a species, we have come far. And gene therapy, in my opinion, is going to be that landmark which is going to redefine medicine as we know it so far in the last century or so. So I think amazing. I could see the response of the public, which was a mixed crowd. Some highly scientific trained people, some from the general public, but everybody seemed to charge up. And personally, I think it was a great motivator for me to continue doing the work, the great work that we here in Novartis are doing.
I think it was really exciting to see how these two parts of the research work together. I mean, the clinical piece and the preclinical piece. These two colleagues demonstrate a marvelous level of collaboration and of competence that I think is really exciting. So my key takeaway is there's a lot going to happen in the field of ophtha over the next few years. There's still a lot to be known, but with people like this who really know what they're doing, I'm confident that we will have a range of new medications that really make a difference in a number of years.
Yeah, this was my second lecture actually today, and I was really impressed about the enthusiasm and the scientific background these two professors from the institute showed to us, and how they were able to combine a difficult scientific topic with, I would say, basic information and convincing people on the importance of this research.